Okay, so the presentation I'm going to do is a little bit different from the normal academic type presentation because Antonio is going to do that tomorrow and talk about some of the basic research. But a few months ago I was asked to put together information that would help people who are out there in the industry, especially those dealing in the meat and poultry part of our industry, and how the research that I've been involved with for the past 20, 25 years, what sort of implications does that research have in the meat and poultry industry in regard to cross-contamination. So this really come about because at this point in our meat and poultry industry, the thought is that one of the major reasons we have a salmonella issue, and continue to have a salmonella issue, is cross-contamination. That they have pretty good control in the animal part, but once it gets to the processing facility, you get this contamination between animal carcasses. So what is the role of biofilm or attachment in this cross-contamination? And that's what I'm going to be talking about. So the main topics are going to be related to attachment, difference between attachment and biofilm, looking at attachment to muscle and skin, biofilm formation on muscle and skin, detachment, and then more with the listeria is attachment to equipment surfaces, biofilm formation, equipment surfaces. So the first part is really going to be, is work done primarily with Salmonella and E. coli, 0157H7, and the final part mainly with listeria. But I'm going to talk about the listeria stuff only more briefly because Antonio will talk about that tomorrow. So I'll concentrate mainly on the Salmonella work. So I think it's good to get in our minds the differentiation between attached cells and biofilm cells. So if you put bacteria on any kind of surface, some of those bacteria will stick to the surface and it'll take some force to remove them, so those are attached. So generally in the lab, if a rinse of water removes it, we consider them not attached. If you actually have to use some sort of physical force, scrubbing, or a forceful jet of water, we consider those attached. And it's kind of arbitrary, but you got to work with something. And technically, one would say a shear force. So attached cells require a shear force to remove. Biofilm cells, on the other hand, are attached organisms that have multiplied and produced a microcolony, or often something larger. So it's a community of organisms on the surface, and you have a community because it grew. So a biofilm are simply attached cells that have undergone some multiplication and released their products, usually a extracellular polymeric substance, as part of this growth. So pretty much any cell that's attached and starts to grow on a surface is going to produce extracellular polymeric materials we call EPS. And so EPS is part of pretty much every biofilm. Now there are some people that maybe think, well, I have a biofilm and I don't observe any EPS. I generally interpret that as, well, you just haven't used the right methodology to observe the EPS. Just because you haven't observed it doesn't mean it isn't there, because EPS is such a widely varying substance. It can be protein, it can be carbohydrate, it can be lipopolysaccharide, it can be nucleic acids. It can be any kind of biopolymer. So you're not always, there's no one way to detect it. The other aspect of biofilms related especially to the food industry is that a biofilm in the food industry is going to contain food components as well as water components, minerals from the water. So we do not have simple biofilms in the food industry. If it's in a poultry processing plant, you're going to have chicken fat on the surface. You can even feel the surface, it's greasy, and you have organisms growing within that chicken fat that hasn't been properly cleaned. And that makes for a very complex biofilm then. In addition, if you have minerals in your water, those minerals will be incorporated into the biofilm, again, making it very complex, difficult to remove. Just some of the basic properties of attached and biofilm cells. They're similar, both slow or no growth, resistance to stresses, and difficult to remove. And I'm sure Antonio will talk about this tomorrow, but the difficulty of removal increases with time, which is really an important concept. So the attached cells, yes, can be difficult to remove, but if you wait around for 6 hours, it's even more difficult to remove. And I'm going to look at this very briefly because is this the same slide you use, Antonio? This is a slide most everybody uses because it's from the Center for Biofilm Engineering, Montana. So the biofilm formation model is the initial attachment that we see there. And attachment can be in clumps or single cells. Sometimes if you see a clump of cells on a surface, you think, it's a biofilm because it must have grown, but cells in the surrounding medium can be clumped and the whole clump can attach. And so you have a clump. It's not a biofilm, it's just an attached clump. But eventually you get the microcolonies with EPS, and they'll grow up and form these larger structures that allow fluids, air to circulate around them, and once they mature, they release the daughter cells, and the baby cells go swimming off. Usually they're motile, and will find a surface and colonize the surface. So this is a whole growth phase of the biofilm. And this is what you would see under the microscope. You see the very kind of diffuse structure you get here when you get that complex structure going up into the air, the three-dimensional structure. So generally the physiology of these cells is closer to that of a stationary phase cell once they're down on this surface. And that also is associated with greater resistance to stresses and chemicals. Yeah, it's a little bit difficult to see here because it's not, but we isolated some bacteria from a water system. It was in a poultry feeding house where the chickens go and get a drink of water. And partially we wanted to see if there's Campylobacter in those biofilms, if there can be. And we actually found that, yes, if you add Campylobacter to these water system biofilms, the Campylobacter will just hang around there, and the chickens could certainly be exposed to that Campylobacter through the drinking system. But these three sort of empty pictures are water system microbes, and this is growing in a low nutrient medium. And it's a pretty sparse type of biofilm they're making. They're just little clumps here, sort of little microcolonies, bigger microcolonies. Note the white area here indicates a three-dimensional aspect. That's white because it's a brighter, more intense color. So it indicates these microcolonies have grown up out into the water. This is actually a Pseudomonas organism. We didn't get this from the water system, but Pseudomonas are typically found in water systems, and this actually came from a surface, equipment surface in a poultry plant. But notice the Pseudomonas makes this really very large biofilm that covers a lot of the surface compared to these water organisms. We can't see it, but we can see there's a diffuse area up here that is probably EPS. So the idea is, I want you to have the idea that biofilms can take various forms, including very sparse types of forms. Going the wrong way. And maybe leave this. You don't have this slide, do you, Antonio? That's from my grad student. But this is different strains of Listeria. And note that the really different structures of biofilm we get on the different strains of Listeria from very isolated microcolonies that are three-dimensional almost, very packed together, to some widely separated diffuse colonies, to just sort of attached organisms with small microcolonies. And here's kind of a fuzzy one, very kind of complete coverage, but there's actually a lot of EPS material in that one. So we could actually looked at, like, I think it was 30 different strains of Listeria and found different biofilm structures. So some of the characteristic presence of EPS make a surface sticky. Well, the EPS protects from chemicals, sticky surface so that organic matter and bacteria accumulate. So once you have your EPS on the surface, other stuff sticks to it. And if you use recommended cleaning procedures, you can remove it. These biofilms are actually readily removed if you use recommended time, concentration, temperature type cleaning procedures, concentration of cleaning agent, whether it's alkali or whatever. So that's just sort of the background information. Now I'll get into a little bit about attachment to meat surfaces. And some of the initial work we did was attachment to chicken skin. And it's actually pretty interesting because you have to look at the microstructure of your chicken skin to see what's happening. And these are just some diagrams here. This is an actual microscopic image, and that's kind of the cartoon image. So your chicken skin has this epidermis area, and then you've got these feathers coming out and kind of a deep follicle here. Underneath you have the dermis and some fatty tissues. And this micrograph shows the feathers in the follicle. And of course, you pull those out, you're left with basically a capillary tube. Now you pull up, so you've got a tunnel in your skin. And when you defeathered the chicken, and the skin contacts water, that follicle acts as a capillary tube. It's going to just suck up that water. There's no way it's not. Oh, nice. So we use confocal microscopy, which gives us kind of three-dimensional images. And it actually allows us to focus down into the skin, focus down into the follicles. And in this micrograph up here, the small spots are the Salmonella, and this is the surface of the skin. It's about the top 5 micrometers. And we can see a lot of Salmonella like lined up in grooves on the skin in there. So the skin is definitely not smooth relative to the Salmonella. It's got this roughness to it. Now if we go and we can actually focus down into the follicle, this bottom image is 30 micrometers below the surface. And the Salmonella are down here. This is the follicle, and the black area here is water, and there's the Salmonella cells. These big dots are actually erythrocytes, so that those are part of the skin cells. They're somatic cells of some sort. Might not be erythrocytes, but these are somatic cells. So the Salmonella, 30 micrometers below into the skin, we focus down into that follicle, we see the Salmonella floating around in there, and actually under the scope you'll see them moving around. Now eventually some of them, and you can almost see it there, a few will attach. So those organisms will then attach once they floated around a little bit. So attachment of Salmonella, and really the type of bacteria doesn't make any difference. They're all going to attach the same. We've looked at different types. As a matter of fact, we even killed them. You kill the Salmonella, it doesn't make any difference. It's going to attach the same. There's no motility required or nothing. So chicken skin, we've got this movement in a water layer on the surface of the skin in those folds. Chicken skin has the cuticle and then the epidermis underneath it. That cuticle is kind of waxy, hydrophobic, and you get less attachment there. You have the folds and feather follicles giving us an initial entrapment, and then you have follicles acting as capillaries that suck in water and bacteria down into that follicle. Of course, if you have those bacteria down in your follicle or in those crevices on the skin, they're released to the surface as the carcass is handled. So you can in fact put a chicken carcass into a bag of sterile buffer and rinse it, run a Salmonella count, put it in another bag, rinse it, and you can if you do that 30 times, you'll find after the 30th time you're still getting some Salmonella off that chicken. Because you can't just get all those bacteria off that skin with one thing. It's a sort of an asymptotic curve, and you never get all the Salmonella off the chicken skin. And there's always something being released. And of course, if your bacteria is in a capillary, a capillary by nature has no fluid flow. So whatever chemical you apply isn't going to get there. Eventually those bacteria in the capillaries will adhere to the capillary wall, and if temperature is right, they'll grow, and you got a biofilm in your skin capillary. And the only way you're going to get rid of it is by cooking it. So any questions on that chicken work? You probably don't want to hear this stuff, huh? But notice,
Try applying some kind of pressure so that you could take out the water in the capillary?
Yeah, of course. And Antonio asks a very good question, because of course you can. And what they did was years ago they used something called trisodium phosphate and these high pressure jets and blasted that stuff at the chicken. And yeah, that helps. But then you've got all your Salmonella out there in the air. So how much has it really helped you? We're talking, now real cross-contamination. So yeah. And note the cross-contamination issue with Salmonella on chicken with the skin is not a biofilm problem. It's an attachment problem. And it's not even so much an attachment problem, but an entrapment problem. And that does make a difference in terms of how you attack it. Okay, attachment to meat. And actually this is work that is done mainly by by Greg Siragusa and Jim Dixon, USDA researchers, because what I did was mainly with chicken, because in Athens, Georgia, we're the chicken people, we're not the beef people. But the attachment to a beef carcass is a little bit different. If you put Salmonella or any bacteria on a beef carcass, you get immediate strong attachment, especially to these collagen-containing structures. And a carcass like this is full of collagen-containing structures. I mean, you know, and I'm not a meat person, but you got fascia, tendons, and connective tissue all over the place. The fascia is that like thin little membrane that covers the muscle. And the bacteria they're glued to it because they're basically strongly attracted to collagen. And it has to do with collagen being more of a hydrophobic protein, right? So bacteria and hydrophobic protein are strongly attracted. But you'll also get some attraction to muscle and lipid. And then you will get entrapment because carcasses have a complex morphology, so you're going to get spaces between muscle fibers and tissues that again can act as capillaries. It's just not as much as hard to deal with as with the chicken skin. But if you have a carcass and cut it so the muscle is exposed, you'll get some uptake between those muscle fibers, but not near as much as you would with a chicken. The key thing is that once it's on that collagen, it's not really coming off. Transferred from tissues by direct contact with tissue, so once you have these pathogens on a carcass, if carcasses are banging into each other, of course they're cross-contaminating. Depends on surface moisture. And this is one aspect of our meat industry in the States is they use a lot of water. Now with beef, it's not so bad. You try to keep the beef carcass dry, and there are stages where it is dry. But the whole idea of avoiding cross-contamination is to keep things dry. Bacteria are transferred through water. And if you have two carcasses bang into each other, they're wet, they're transferring. Aerosol production, because in a meat facility you have physical force being applied to the carcass, generating an aerosol. Transfer of fluids and particulates, and this again can be rinsing carcasses. It can be anything that is generating a wet aerosol or a dry aerosol. Now in our facilities, both poultry and meat, the facilities are kept pretty cold. I'm thinking, I'm not exact on this, but certainly well below 10 degrees C, and I believe below 8 degrees C. And some of them are 5 to 7 degrees C. So I think legally it has to be below 10 degrees, but most of them are kept colder. So you actually have very limited temperature for, like, Salmonella is not going to grow that much at those temperatures. So you have limited ability for biofilms to form. But if you have acceptable temperature nutrient conditions, you certainly will get a biofilm. And the other aspect of attached cells is, even if they haven't grown to produce EPS, they're still going to be associated with organic matter that's going to make them difficult to kill through chemicals. And they're still, even as attached cells, they have resistance to various stresses. Now, kind of summarizing this attachment biofilm progression for meat association with superficial water. So if you have, whether it's a chicken or beef, whatever, and it's been rinsed, you're going to have a thin layer of water on the surface, and these salmonella are going to be kind of moving around in that thin layer. And I know that because we look at it under the microscope, we can see them moving around. And this is, you think, well, they're moving around. Well, this is after we rinse it. So when we do our research, we put the salmonella on the chicken skin and we rinse it. And then we look at it under the scope, and we'll see the salmonella moving around in that thin layer of water. So I mean, that's. Then you're going to have uptake into pores through capillary action. You're going to have reversible association with surface tissues. This just means it's not very firm. You could easily remove it. But that will, over time, go to non-reversible association because you're going to get hydrophobic and electrostatic bonding. Some of this through the EPS the bacteria produces. Then you're going to get production of EPS, then penetration to subsurface over time. But by the time you get penetration, well, that's actually, you could call penetration to subsurface being taken up by capillaries. This is going to depend specifically on the tissue. But over time, you're definitely going to see more of the bacteria at the subsurface. And by subsurface, I'm defining that as more than 5 micrometers below what we focus on. I mean, there's a microscopic subsurface. But we've actually been able to see salmonella 120 micrometers below into the chicken skin in a feather follicle. Okay, that's a major subsurface. That's a major follicle. So steps 1 and 4 occur rapidly. These kind of reversible to non-reversible through hydrophobic and electrostatic bonding. And that actually, when you're looking at attachment to collagen, that's your collagen attachment. So that by rapidly, I mean within 30 seconds or so. I mean as rapidly as we do the experiment. EPS then is going to take longer time. Step 2, well, that requires water. Step 5 and 6 take hours to days. So 5 and 6 here, this non-reversible association and some penetration depend upon time-temperature associations. And unless you're looking at capillary uptake, which is pretty immediate when you hit it with water. But this EPS production maybe 4 or 6 hours. I don't think anybody really knows for sure how long this takes because it's very hard to measure that on a meat surface. I mean, I don't have a way to do it, to observe that on the meat surface. But certainly if it's kept cold, it's going to take many, many hours for that to happen. And so maybe days to hours. Bacteria become more difficult to kill or remove as attachment proceeds. So as you go down from 1 to 7, you're making things much more difficult to reverse, and you get to the point where only cooking is going to help you. Practical implications. So my advice out there in real world is rapid removal of contaminants is the most important aspect. So if you can't avoid having, in some cases, fecal contamination or where you know there's some contamination happening, and you have to be able to rapidly remove that before it gets down into the subsurface. Make sure clean water, of course, is critical. Skin surfaces exposed to water should not be contaminated. So certainly in poultry, we have our poultry with Salmonella contamination on the skin and then having it going into a chill tank, which is bad. In Europe, they'll use more air chilling, which is much, much better, I think. Chemical interventions are effective when applied immediately after the contamination event. And this actually, there is really good evidence from the industry for this, because in the States we have various chemical interventions that are used, things like a dip in peracetic acid, for example. And you have to do that kind of intervention immediately after the contamination would occur. And we're talking minutes, not 30 minutes, but 2 minutes, 1 minute, or just as quickly as possible. And when they set up the lines in the plants, they have observed that if they do their intervention further down the line, it's much less successful. You have to do it right away. So that's it for the meat. I'll talk a little bit about equipment. Any questions on the meat stuff before we go to equipment? Antonio has no questions. So the thing that surprised the sanitarians I was presenting this to is that the whole meat thing is not a biofilm problem. But if you've got a biofilm on your meat, you got spoiled meat. Okay, so in the industry, practically speaking, there should not be any growth, and that's because we keep things cold, is mainly. And if you have pathogen contamination, you also have Pseudomonas, you'll have a slimy surface. So, equipment, though, we have a little different situation. We do have ample sites for attachment. We have, where's my, I know, I got it. I got to press another one there. There we go. That's, my student put that in there, I guess. So here's some stainless steel. That's our micrograph of stainless steel. Actually, it's not a micrograph. That's a confocal laser micrograph. You can't get that with electron microscopy. Now you can see it's stainless steel, and this is food grade. Has a lot of grooves in it from the mechanical polishing, and sometimes the mechanical polishing is not perfect, so you get these sort of rougher areas. So it's certainly not smooth. It's charged, slightly hydrophilic. And we're looking at attachment to such a surface. We'll get an initial reversible association on a conditioning film, which is just an organic layer that forms that's attracted to it. Then we'll get the stronger bond and the EPS materials. This is what I'm not going to talk about, because Antonio is going to talk about that tomorrow. He might not tell you, though, that the smoother the surface, the less attachment you get. So, and one can say, well, why don't we use really smooth surfaces? Because you can get something smoother than stainless steel. You can get, for example, Teflon that's really, really smooth. And the reason we don't use smoother surfaces because they don't stay smooth. They get scratched or whatever. And then that scratch becomes like a bacterial magnet. So it has to be smooth, but it also has to be able to stay smooth. Okay, well this one we can see, so we envision that stainless steel with the polishing grooves. Here's the Listeria biofilm growing in those grooves, and that's a pretty good biofilm there. And then here's another Listeria biofilm, and we can see the water channels in that biofilm, and there's a groove. That's kind of hard to see in this light, unfortunately. But it is growing in a grooves down there. You can barely see that. So some of the practical aspects: daily cleaning prevents formation although it may build up during downtime, more often you're going to get these biofilms on sites that are not clean but still get wet. And so your main factors are water and cleaning effectiveness. So if you have a surface wet, you better have effective cleaning. And floor drain surfaces, those are all places you'd get it. Looking at detachment of cells. Are you going to talk about this tomorrow? No? You're not. So I have to talk about attachment then. Cell-directed. So the biofilm itself, the cells can make enzymes that release themselves. Spontaneous release, just as the cells grow, they're just flying off into the liquid. But then, of course, there's external directed, where one can use a scrub brush or other shear force to detach, as well as chemical detachment, oxidants, surfactants, chelating agents, enzymes can all be used to detach cells. Chelating agents, because this attachment often has calcium bonds helping to attach it. So, the importance of attachment versus biofilms, and now we're talking about the kind of equipment surfaces you see in the processing facility. And of course, the biofilm requires time and temperature, and one should hope that one can control the times and temperatures to avoid the biofilm buildup. But our equipment surfaces are always going to have the attached bacteria sitting there ready to grow. So presence of biofilm, it's poor equipment design or poor cleaning procedures. I think it's nearly always one of those two. I'll show you the data soon. And avoidance of biofilm is important because it provides conditions for pathogen growth and survival. And these sites where these organisms are will provide a continuous source of pathogens going into the processing facility. So in our meat processing facilities, we're looking at good design and effective cleaning, good design, cleanable surfaces, no dead ends, easily disassembled equipment. What's a dead end? A dead end is a place where there's no fluid flow, but it's wet. So a macroscopic dead end is a pipe that's sealed off. But usually these are microscopic dead ends where there's like a crevice or a crack or something. So fluid can get in, nutrients can get in, but there's no cleaning solution isn't going to circulate there. So it's a dead end relative to the bacteria. Easily disassembled equipment, anybody that's been working in a meat plant knows that a lot of the equipment is very difficult to disassemble and clean, and so it doesn't get disassembled and cleaned very often, and that's why they have problems. So that's an equipment design issue. Effective cleaning, how often are they going to clean equipment? And that's a question I get asked a lot because I give extension workshops to our industry folks, and they always say, well, how often do I need to clean this? And the answer is, well, as often as it needs it. The only way to know is to do bacteria tests and see if it's often enough. So you get some data, then I'll tell you if you have data. Using recommended cleaning agents, and cleaning, not sanitation, as primary control. So if you use recommended temperatures and recommended concentrations of cleaning chemicals, and you don't have dead ends, you have good design, the biofilm will be cleaned away. So cleaning is your primary control. If the equipment isn't cleaned, it's hard for the sanitizer to actually remove it. That's why I say cleaning is the primary control, because you want to remove the food nutrients with the cleaning procedure, where sanitizers don't do that. So we tested the ability of some cleaning procedures to remove biofilm polymeric substance. This is EPS produced by a Pseudomonas organism. So there's some sodium hydroxide, it's 66. some at 70 degrees. That's the control, no treatment. So that's the EPS without treatment. This is our 1.28 for 3 minutes, is not very good at 66. We go up to 70 degrees with 1.5, it's pretty clean. So adequate temperature time cleaning is important to remove that EPS material. And there's a lot of food facilities will not reach 66 degrees in their cleaning. And they'll tell me, oh, no, of course not. We never get it above 60, they'll say. Well, that's what they're getting then. Now you cannot compensate for low temperature by increasing chemicals very much. A little bit, yes, but ultimately you have to have some temperature. And that's like the biggest factor, I think, in biofilms on equipment is that the companies are not getting their water hot enough to clean the equipment. To me, it's just outrageous that they're not doing that. You got to have hot. I mean, everybody knows you got to have hot water to get something clean, right? Everybody knows that. Why do they think they don't need hot water? Oh, it's just warm. Okay. Controlling it. We use aerobic plate counts, ATP tests to detect, and then investigate the cause. If you get high counts, you can figure out why do you have a high count. And differentiate operational causes from design efficiencies. That means you're going to have to determine, is the reason for the high count that my cleaning crew isn't doing a good job, or is it because they can't do a good job because the design of the equipment is too terrible? This is some data we collected. These were painted concrete drains. That's before stainless was concrete. 292 million aerobic plate count before, 167 after. When they installed stainless drains, 197,000 before, 700 after. So by having a cleanable surface, the cleaning crew could do a good job and effectively clean it. Of course, that was expensive to have a cleanable surface, but in this case, the company found it worthwhile because of the drains were very close to the product.
So, Professor, I'm sorry, what differs there is the material surface?
That's the material, yeah.
Because the reduction is the same.
Yeah.
The initial count, it's different depending on the
Yeah.
material.
Yeah. So this is just a painted surface. It just builds up the biofilm. And once you build up the biofilm, they can't get rid of it adequately. So paint is, everybody knows this too. It's hard to clean a painted surface. Stainless steel, much easier. So the biofilm doesn't build up on the stainless steel, and then of course you can clean it.
Professor, may I interrupt you?
Sure.
Some days ago, I was talking about our work, and there was somebody in the audience, some person that was a specialist in materials,
Mm-hmm.
and he asked us, what about founding another type of surface material to replace stainless steel?
Mm-hmm.
What kind of characteristics should
Yeah.
the material have to avoid?
Yeah, they've developed many types of materials, including materials with coatings.
It's not very
Yeah, the problem has always been durability. So you purchase equipment and you spend $200,000 on this equipment, You want it to last a number of years to get your investment back. And if you make it out of like, if it's the material's coated, that coating comes off. But also, even if you put like silver impregnated into it, if the organic material coats the silver, the biofilm grows on the organic material, it doesn't work. So it's not as easy as it sounds. But the main thing is stainless steel is durable, whereas many of these other materials just aren't. I mean, it has to be resistant to all the cleaning chemicals, resistant to corrosion, resistant to scratching. Very hard to find materials that will do that. And the other thing is, I worked with a company that actually had a material that was very smooth and very durable and very easy to clean. If the biofilm was on there, you just took a hose and rinsed it off because it never stuck to it. Wonderful surface. But it cost 5 times as much. So nobody bought it. You got that? Yeah. So you got to have it. It's durable, but something people can afford. So yeah, it's good. Time for the summary. So microbial contamination, cross-contamination of meat, primarily an issue of attached and trapped cells. Longer time is an important aspect there. Bacteria are internalized in meat tissues, especially through water uptake, where capillaries associated with the meat suck up the water, and you can't get rid of them or kill them until you cook the meat. Recommended cleaning procedures will prevent biofilm formation. Because I get asked, I said, we have to improve our cleaning procedure. We need better chemicals. Generally, the answer is no. The chemicals work fine. The reason you have contamination issues is because of poor design or poor temperature and it's operational types or design types of things that I talked about. It's not because of the chemicals. But that hasn't prevented the chemical companies from coming out with new and improved chemicals. It's just, that's not really the problem, but they make a good profit off of them. Disassembling might be required, and nobody in the industry wants to hear that they have to take stuff apart to clean it, because it's hard to do that and it takes time. And some of this equipment is actually pretty dangerous when you get some of these meat cutting types of machines. They can slice your finger off if you're not careful. They're so darn sharp. Proper temperature, cleaning agent concentration. Biofilm problems caused by poor equipment design, poor structural design, lack of equipment assembly, lack of effective follow-up to lab results. I put that in there because I have talked to people and been to facilities that have been involved in recalls and even outbreaks, and much of the time it was lack of follow-up to lab results, where they were getting positive lab results. They were getting lab results telling them their equipment, their environment is not clean. They were getting listeria coming up positive, and they simply did not know the proper actions to take. Or they thought, because I'm only getting listeria 1 out of 5 samples, it's okay, sort of under control. They'll say, well, we're taking samples every week, and every 3 weeks we see a listeria, so it's not so bad. Let's not do, we don't have to do anything. So it's those types of approaches that have led to recalls. And if you're wondering why that approach doesn't work, I have to get into the statistics of sampling. But what it has to do with is the statistics of sampling and figuring out, that a positive sample every 3 weeks can be statistically significant, right? Just talk to Cadbury with their Salmonella recall. I believe that was an issue of not looking at the statistics of sampling. Okay,
Professor, may I interrupt?
sure, I'm done, so you're not really interrupting anymore.
I was thinking, so you said that cleaning is very important. And you also mentioned that temperature is very important.
Mhm.
And regarding to the disinfectants, in their ability to eradicate biofilms, there are any active substances that are better than others?
Yeah, so if one has a complex piece of equipment, that one trying to clean and sanitize, if you have it cleaned properly, so you've removed the food residues, any sanitizer will kill the remaining organisms, any of them. But if you don't have it effectively cleaned, you have some organic residues remaining, then you have issues where a sanitizer like the peracetic acid will be more effective because it works somewhat better in the presence of organic matter than other sanitizers. And even quaternary ammonia compounds. But we don't, quaternary ammonia compounds are not approved for food contact. So peracetic acid becomes a good one. But you have to be careful because if the companies will go to peracetic acid, but they won't look at the reason why that equipment wasn't getting clean. And so if you just decide, well, I'm not going to worry that my equipment wasn't clean, I'm just going to throw a lot of peracetic acid at it, then you have a piece of equipment that is building up organic residues somewhere, which you haven't figured out where or why. And eventually the peracetic acid is not going to work because you're going to have this, a true slimy biofilm there somewhere that you haven't figured out. So I always try to get these people to figure out why the equipment isn't getting clean and say, please concentrate on cleaning. That's just like the heat. I did a study, one of my students got her doctorate degree, and actually, there's my acknowledgments here, and she's not up there, but Revis Chemielski did her doctorate degree on killing Listeria biofilms with heat. And I did this because this is what people in the industry wanted to do. But in fact, if you haven't cleaned your surface and you use heat, you will cook on the dirt or the soil, and you'll have a buildup of cooked on soil. So I don't recommend using heat to do that either. But we do have companies that have complex machines. They'll run it into an oven and cook it. Cook their machine to kill the listeria. And they're cooking all the soil, all the food that they haven't removed by cleaning.
It's possible to remove all the water to dry the equipment.
But it's not. Some equipment is simply designed so that there's pockets of water. Yeah. And that's why you have poor design, or you have to take it apart to drain it. This is a big problem. Yeah. And if you have that, then of course you can put your peracetic acid in there and have acidified water, but now are you corroding? What's that acid doing to your equipment? And you put chlorine in there, chlorine's going to corrode it too. So you have problems with acidification, but that's better than not, I guess.
Frank, I was thinking, you were saying that the purpose of applying the temperature it is important, right? But I'm just thinking, what if it's a really big equipment?
Mm-hmm.
You will have water at 100 degrees, let's say, in the beginning of the equipment. You can't reach it by the end.
Exactly, that's exactly the problem.
Temperature, would it be any good to use steam or something?
Well, perhaps, but you can use, you should use water starting at 80 degrees, and by the time it's done, it's at 66. That's the way you should work. except the company you're working for probably doesn't have water at 80 degrees. They can't make it. That's the problem. You know, the guys will come back to me and say, well, our boiler room will only give us water at 66 degrees.
Because I was thinking that they had problems at getting it at a good temperature by the end of the cooking.
Yeah, this is true. You have to start higher than what you need. Yeah. And you have to be careful because if you start too high, you will cook on the proteins. If it's protein, especially milk. So if you start your milk at 80 degrees, your milk residues at 80 degrees, you'll cook on the milk protein. You have to start your milk with like a 60 degree rinse, and then go up higher after you're rinsed off yeah. And
So, do you have questions?
any other questions? So I hope you appreciate some a practical application of some of what was fairly basic research that we did. Some of this going back to the 90s, going back quite a few years, but I was glad to be able to get it together into this presentation. Thank you.